So, children, now you are seeing two diagrams on the screen, and the heading is a structure of a blastocyst. First of all, you should be realizing again that what is the structure of blastocyst looks like, and then how it helps in implantation. I will be using many technical words during the explanation, so please take care. First of all, we will be talking about this structure. Right, this structure you have already drawn during the embryonic development of blastocyst. Uh, you just see that the outermost layer is actually red colored, and this is expressing zona pellucida, right? And then you know already that there is the trophoblast, the, the inner layer of cell, single cell layer, that is trophoblast. The cavity is known as blastocoele, and the inner cell mass. Now this inner cell mass, yes, usually in my last video I used the word embryonic knob, or you can also call it embryoblast, when you have to divide all the cells of morula into two categories. The outer one acting as a covering, it is trophoblast, and the inner one making a knob is embryoblast, right children? And the cavity is blastocoele. I believe that after seeing this diagram, now you will be able to realize what was the structure of the blastocyst. Now, if you can see this arrow indicates that this blastocyst is going for, now it is supposed to go for implantation. Right, children? So during implantation, what should be the first thing to be happened here? Just now in my previous video, I told you that till the time this zona pellucida will be there, there will not be any sticking on the wall. So that means the first thing which is to be done here is the removal of zona pellucida, and this removal is done by the proteolytic enzymes which are secreted by the trophoblastic cells which are located just beneath zona pellucida, right? So proteolytic enzymes which are secreted by this trophoblast actually dissolves zona pellucida, therefore there is no outer covering now present around trophoblast, right children? Now this trophoblast is absolutely ready for implantation. Understand, by this time you have the idea that trophoblast contains enzymes. These are lysing enzymes, or you may say dissolving enzymes, right? Fine. Now you just refer to this diagram. After the arrow, during implantation, what happens? So zona pellucida is gone, and now you have to talk about these two layers, if you can see, right? Just in my previous video, I used that word division of trophoblast. So if this trophoblast is a single cell layer, now it will start dividing and it will form a multicellular layer, right? I mean, it's a 3-4 layered structure then. Out of these layers, few will be towards the inner side during the division. Now you understand periclinal division, though we cannot use the word here, but alignment could be understood that if there was one cell here for trophoblast, when it divided into two, one cell would come towards the inner side and one towards the outer side. That's what is happening here. This layer is inner one and this layer is outer one, right children? So this is what I meant to say, right? So trophoblastic layer has divided into a bilayered structure. As of now you can see a bilayered structure. So the inner one is cytotrophoblast. This is how you'll be able to see it. See, cytotrophoblast, the inner one, right? That means when the embryo would be forming inside, so this is the layer which is in direct contact with the embryo. So this is inner one, right? You call it cytotrophoblast. And the outer one, which is making a continuous wavy line here. You can see outer one is wavy, and the point of contact. Now always remember one thing here, that when the blastocyst settles on the endometrial wall, the layers, the trophoblast, it always ensures that this implantation should occur in such a way so that the embryonic pole or embryonic knob should be touching, right? So this should be the point which should be here while while the blastocyst is settling over the endometrial wall. Embryonic knob, or you may call the embryoblast cells, wherever they are attached to this trophoblast, they should be touching this side. Just see the alignment and understand what I'm trying to say. Right, children? So later on, you can expect that during development, the point of contact would become this, and then the placenta and the fetus. This is just to give you an idea. Therefore, the cells are supposed to be here so that further they can develop. Right, children? As of now, that explanation is not needed. You just understand that trophoblast is divided into two layers: inner one, cytotrophoblast; outer one, syncytiotrophoblast. Here, and I have written in within the bracket here that syncytiotrophoblast, which is going to touch the endometrial wall directly, is going to form the future placenta, and cytotrophoblast, which is in direct contact with the embryo, this is going to form the protective embryonic membrane, chorion. In future, this inner one would be forming chorion, and the outer layer of trophoblast would be forming placenta. Now let me tell you, every time when we are saying placenta, what do we exactly mean to say? Children, after seeing this diagram, you can see that this outer layer is actually trying to intrude into the endometrium by forming villi. Yes, these finger-like projections are villi. It is trying to intrude into the endometrium. These red cells are endometrial cells, and it tries to displace certain cells. You call it DCR. Now this is decidua cortical decidua cell reaction. Decidua, why you are calling it decidua? Because the moment this implantation occurs, endometrial cells are known as decidua. So since these cells are supposed to be displaced or degenerated, so that there could be some space for this outer trophoblastic layer, syncytiotrophoblast, that's why you call the reaction as DCR, right? When I am using a word villi, right, these finger-like projections are villi. If you can see, when you would just open your palm and you try to interdigitize, you try to intermingle your fingers, the term is known as interdigitization, right? Similar is the case here. You can see that the endometrial line, when this outer trophoblast is forming the villi, there are the structures in between which are endometrial structures, right? If you can let me make it very clear, these structures are endometrial structures, and if you see from the other side, these are also villi which are from the maternal side. So understand now that there is this structure now is known as placenta. You know why? Because some contribution is from the embryo side, you will call them chorionic villi, and some contribution is from the maternal side, so you will call it placental villi, right, or endometrial villi. But ultimately, wherever there is a point of contact and that tissue is quite spongy, quite vascular, you call it placenta, right, children? One more thing that I had to tell you was, about syncytial trophoblast. Now this was about syncytial trophoblast. Better I should use some other color, otherwise you will get confused. Just hold on. See, if you can see the inner cells, they have cell wall, cell membrane. Why I should use the word cell wall? Cell membrane, and then the nucleus. But in the outer structure, the cell membrane is not actually visible. If you can see the nucleus, all the nuclei, they are freely suspending in the villi, and the cell membrane is indistinct. That's why you are using a word syncytio. If you remember when you studied free nuclear endosperm, we use the word coconut syncytium. That means a liquid in which which was multinucleated. Similarly, here you can see a structure where cell membranes are not there and the structure seems to be multinucleated. Right, children? So again I am taking a recap that during the settlement on the endometrial wall, take these cells as endometrial cells, and the blastocyst is going to settle over. But the first thing that is going to occur is the removal of Zona pellucida. It has to go off, and after that the trophoblastic layer has to divide into two. The inner one is cytotrophoblast, outer one is syncytiotrophoblast. Right? Cytotrophoblast would be forming chorion so that the inner embryo could be protected, inner fetus could be protected, and syncytiotrophoblast is going to form these villi so that the placental formation could be worked out. Right, children? I hope this much is clear. Just adding to my point is that since these outer syncytiotrophoblast is going to form the future villi, future placenta, in that case, this is to be discussed now. I have used this word in class 11th lot many times, even in class 10th too. HCG. HCG is human chorionic gonadotropin. Right, children? And HCG is secreted. It does two works basically. First thing is it ensures that the corpus luteum, because now fertilization is done, so in that case corpus luteum has to persist till 9 months. So in that case it ensures that corpus luteum stays for 9 months and progesterone level is maintained. These two functions are there for HCG, and HCG is a placental hormone. Right? I told you earlier as well, again I am letting you know that HCG secretion is a confirmation of implantation. That is why when a lady conceives, the urine test is done to confirm whether HCGs are there in higher concentration or not. If they are present, that means that the placenta has been formed. And all this implantation, all this procedure, this is completed by the second week. 1 week is taken for the blastocyst to come to uterus, and by second week all these implantation steps are completed. Right, children? So just go through the explanation once again, see the videos as many times, but you have to make this clear. Right?